Unit 2 is cell structure and function. So in this unit, you're going over everything about cells, what they're made of, how to tell different types of cells apart, the function of different organelles within those cells, and just everything that really starts you off with all of biology, because all living things are made of cells. So biology is the study of living things, and since excuse me, since all living things are made of cells, then it's really important to understand uh, how these cells work, what you're going to find inside these cells, and everything that we do as living organisms is dependent on all of these forms of work that cells do. And all of the pieces that go into how they perform these jobs, like building proteins and breaking down sugars and these different activities that your cells are doing, uh, is going to be determined by uh, the different organelles within those cells that participate in those jobs. And as we go on to future units, you're going to see that we come back a lot to Unit 2 because you need to understand um, what organelles are involved in something like the activity of enzymes, which is going to be Unit 4, and the processes of photosynthesis, which is going to be Unit 5, and metabolism, which is covered in Unit 6. And so all of these processes are involved in this work that is done by cells. And so making sure you're really clear on the different organelles you need to be familiar with and what they do and what they look like so you can identify them, and all of these structures within cells is going to be really important going forward. And the first exercise, so when you're in exercise 1 and you're taking measurements of different cells, we're first looking at one of the biggest divisions in cell types. And that division is between prokaryotic and eukaryotic cell types. Now, those names refer to um, essentially the type of organelles you're going to find within those cells. And the prokaryotic ones have no membrane bound organelles. That's an important distinction because they still have some organelles, just the ones they have do not have their own membranes. That means that there's going to be no nucleus. The nucleus is an organelle with a membrane that contains the DNA. Now, they still have DNA because that DNA is the building instructions or code or that determine everything else that becomes part of that cell. It's just not found within a nucleus. It's free floating within the cell. And they're not going to have things like mitochondria or chloroplasts or any of these other organelles that you'll end up looking at that are within eukaryotic cells. And because they have none of these membrane bound organelles, they are extremely tiny compared to eukaryotic cells. So prokaryotic cells are very, very, very small. And the kinds of organisms that are going to be prokaryotic are going to be bacterial cells and blue-green algae. Blue-green algae is another prokaryotic type of cell within, so both of these make up what we call kingdom Monera, which you'll learn more about in Unit 9. There's some other organelles, or not organelles, excuse me, there's some other organisms that are also prokaryotic, but they're part of another kingdom that we don't learn about in this class. If this kind of stuff interests you, you'll want to get more into a cell biology course where they'll talk more about those kind of organisms. But the main ones you'll be concerned about are these bacteria and blue-green algae, which are two different types of organisms found in Kingdom Monera, and all organisms in that kingdom are prokaryotic. Then your eukaryotic cells have all of these membrane organelles that we said prokaryotic cells do not. And those membrane-bound organelles take up some space. And because they take up some space, those cells have to be bigger. They have to be big enough to contain those organelles. And a major theme in this unit is that smaller is better. The smaller you are, the more efficient you are, because you increase your surface area to volume ratio, which we'll talk about a little more in a future slide. But that means these prokaryotic cells can be very, very efficient because of how tiny they are. But another thing that helps with efficiency is specialization. When you can specialize for a particular job, you're more efficient at that job and you can produce things a lot faster. And we think of that as a good comparison for that is factories, because in a factory, every individual worker there or every individual machine within the factory that's contributing to the production of whatever product they're churning out. You could say a candle factory, for example. If you're making candles in the candle factory, one person who works at that factory isn't going to do all the steps involved in making the candle. They're going to have one specific job that they are specialized to do. Something like cutting the wicks on the candles. And because that's the only job you do, it's super, super simple. You just stand there and you have scissors and you cut the wick to make sure it is the length it is supposed to be. And that's all you need. You don't need to do anything else involved in making those candles. You don't have to do the formula for the wax. You don't have to glue the wick to the bottom of the candle. You don't have to package it. So you don't need tape and you don't need boxes and you don't need any of these other stuff that anybody else in the factory needs. And eukaryotic cells work that way compared to prokaryotic cells. Prokaryotic cells are super, super tiny to increase their efficiency, but eukaryotic cells specialize and break down jobs to become more efficient. And that's what all these organelles do inside there. The organelles are like the individual workers that have these very specialized jobs that help this cell to be more efficient. So it's essentially two different strategies for efficiency. One is be as small as you possibly can so that you increase your surface area to volume ratio as much as possible. The other is having organelles that specialize in things to break down tasks to help you turn things out more efficiently. And so in this image, we have some things that are shared between these cells, and these are going to be traits or things that you'll find in all living cells. They all have DNA, all have ribosomes and cytoplasm and a membrane. And then prokaryotic cells have some unique traits to them, and eukaryotic cells have some unique traits to them. So that's exercise 1 gives you a real good look at these two cell types and some differences in size that become really important in those cell types. And then in exercise 2, excuse me, you start looking at lots of different organelles that are found within these eukaryotic cells. And I've got an image here of a specific type of cell. This one is of a neuron. And I like this image because it clearly identifies a lot of these organelles that you need to be familiar with. And you can go through and identify some key features that help recognize those organelles. So your nucleus is nice and clearly identified here. And in that nucleus, you're going to have your DNA. That's those instructions or that blueprint for everything else that happens in your cells. And you don't see it in this image, but another organelle you need to be familiar with is the nucleolus, which is actually within the nucleus. And in the nucleolus, you have some tightly packed DNA that it will come out into the outer covering of the nucleus when it's essentially being unfolded so that those instructions can be read to start doing something like transcribing RNA that will then come out of the nucleus for making proteins and other things that are needed in the cell. So you don't see the nucleolus in here, but that's another one you need to be familiar with, and that nucleolus is within the nucleus. Then we have some rough and smooth endoplasmic reticulum all around our nucleus. This can be attached to the nucleus essentially, and you'll see squiggly lines coming off the nucleus, or it can be floating around within the cytoplasm of the cell because it's going to be taking the proteins or lipids that it has made somewhere within the cell. So the jobs of the rough and smooth ER is specific to which type of ER you're talking about. Rough ER is going to be making proteins, and smooth ER is going to be making lipids. And you can tell them apart because the rough ER will have some stipulation on it. It's got little dots all along it. Those little dots are ribosomes, which you also should recognize. It's one of your organelles you need to know for this course. And ribosomes also help with making proteins, just like the rough ER does. So they are on that rough ER, also assisting with the making of proteins. And ribosomes can also be on their own all through the cytoplasm. So you'll have rough ER with ribosomes making proteins, and you'll have smooth ER without those ribosomes. So it's smooth because it doesn't have the little dots all over it, and it's going to be making lipids. Then we also have identified in here a Golgi apparatus, and it's got this kind of flopped over look with little bulbs coming off the sides. One description I've seen for it that really helps me recognize it is a stack of pancakes that has had syrup poured on it, and so those little blobs are like the syrup. But if you have something else you can think of that this looks like that helps you recognize it, that's good too. And the Golgi apparatus is something that helps essentially package things. And by packaging, it's getting it to bend and conform into the shape it needs to have. And shape is also really, really important in all the various activities that happen in our cells, because being a certain shape will determine how well you can match up with other things of certain shapes, or whether you can get into or out of the cell. And lots of these interactions are very, very dependent on your shape. So it helps package things into the proper shape and get it where it needs to go. Then another one you need to recognize here is the mitochondria. So that's these here. And this will always have some kind of rod shape or bean shape to it, with lines going through it that the text describes as finger-like projections. I think it looks kind of like a maze or like a zigzag going through it. Here's another one with the zigzag. And that's the outer and inner membrane. Those zigzags are the inner membrane of the mitochondria, and it is the powerhouse of the cell. They call it the powerhouse because it's where it makes the energy that your cells are going to use. So all of the processes in your cell that require energy use it in a very specific form. That energy has to be ATP. And so mitochondria take sugars and turn it into ATP. And that happens in reactions that happen across this inner membrane. Membranes are composed of lipids and proteins that are interspersed between the lipids. And those proteins work as channels that things can pass through. And sometimes those proteins also help speed up reactions. So those proteins within the inner membrane of the mitochondria are going to be ones that are helping catalyze these reactions that turn sugars into ATP. So it's taking sugars and making the energy that the cell needs. Then we also have identified here the membrane, which is the outer portion of this cell. And as I mentioned, those membranes are made of lipids and proteins, and it's what we call a semi-permeable membrane because it allows some things to pass through it and other things to not pass through it. It's also what we call fluid-mosaic. That means that the pieces that make up the membrane aren't rigid. They don't stay in one place. They can move around. And because they can move around, it's like a fluid almost. And it's mosaic in that it's all connected and intact, but it's not rigid solid. It kind of lets things move around. So that's all of the labels that you should be familiar with from this image after going through the lab. And all of these organelles here are going to be found in animal cells because a neuron is found in a nervous system, and nervous systems are going to be found in animals. So there's some other cell features you'll need to recognize that are in plant cells that are not in animal cells. And that comes up in exercise 3. So in exercise 3, we look at some similarities and differences between animal and plant cells. So some of those similarities are going to be all of those organelles that we just talked about in the last slide are going to be organelles that are in both of these cells. Both plant and animal cells have cell membranes, and they have a nucleus and nucleolus, and they have mitochondria, and they have Golgi bodies, and they have smooth and rough ER. Now, what the plant cell has that animal cells do not have is they are going to have a cell wall in addition to their cell membrane, and that cell wall is an extra outer layer that is going to be more rigid, and it helps plant cells keep their shape. And this works together with another unique feature in plant cells called the large central vacuole to create what's called turgor pressure. And turgor is a fancy science word for water, so we're really talking about water pressure. And that water pressure is because that large central vacuole fills with water. And when it fills with a lot of water, then this large central vacuole will take up most of the space in the plant cell. It'll push all the organelles to the edge, and it will be filled with mostly water in that large central vacuole, and it pushes against the cell wall. So when the large central vacuole is full of water and pushing against the cell wall, that creates turgor pressure that keeps it nice and rigid and strong, so it keeps its shape. And the large central vacuole is unique to plant cells, but both cell types, animal cells and plant cells, will have other vacuoles. So there's a little vacuole here in this plant cell, and there's a vacuole here in this animal cell. They'll both have these vacuoles, and the job of all vacuoles is storage. So that large central vacuole in the plant cell strictly stores water, while other vacuoles will store more water, or they might store some waste, or they might store some of those sugars or other molecules that are going to be a sort of food source for other organelles within the cell. And so they store things. And then the last thing that's unique in plants is chloroplasts. So chloroplasts are going to look something like this in your pictures. They'll always be this kind of round or now I'm forgetting the word, not quite round, but like oblong kind of shape with something that looks like stacks inside it. That's that inner membrane on the chloroplasts. And you can describe it as a stack of coins or stack of pancakes. Just don't mix that up with your description for your Golgi body, which is like a sloppy stack of pancakes with syrup all over it. This is a nice solid stack here. So those chloroplasts are unique to plant cells because they're involved in taking sunlight energy that's in wave form and converting it to sugars. And animal cells don't do that. That's a process that's part of photosynthesis. And essentially, they're making their own food source out of that sunlight energy. And animals don't do that. We don't make our own energy. We have to ingest it. And we ingest it every time we eat something, whether we're eating plants or other animals. But every time we eat, we are ingesting particles that will then get broken down and turned into our energy source. So plant and animal cells are both eukaryotic cells. Remember from exercise 1, eukaryotic means it has membrane-bound organelles inside. And so all these membrane-bound organelles mean we're talking about eukaryotic cells. And they're both going to be cell types found in multicellular organisms. Animals are all multicellular, and plants are all multicellular. And then they're going to share a lot of organelles that are common between them, but the plants are going to have a few unique ones. Their cell wall is unique, their chloroplasts are unique, and that large central vacuole is unique. So then in the next exercise, exercise 4, you start looking at some differences between single cellular organisms and multicellular organisms. Now, in exercise 1, we already looked at some single cellular organisms with the prokaryotic cells. So all prokaryotic cells are going to be single cellular organisms. So that bacteria and blue-green algae you should be familiar with are going to be single cellular. That means one cell is the whole living organism. A bacterial individual cell, that's it. That's the living thing. But There are other single cellular organisms that are eukaryotic. There's going to be some we call protists, and they are eukaryotic single-celled organisms. So they're going to be different from the prokaryotic ones because just like the plant and animal cells, they have membrane-bound organelles. That's why they're called eukaryotic. But one individual cell is the whole living organism. And you looked at one of those in the first exercise with what you took measurements of. The second image that you take measurements of was of a paramecium, and a paramecium is a single-celled eukaryotic organism. So if you're looking at strictly eukaryotic cells and you want to compare a single cellular eukaryotic cell to a multicellular eukaryotic cell, you will find that the single cellular ones tend to be a bit bigger than the eukaryotic, the multicellular ones. And they tend to have more excess organelles. Now, the reason for this is single cellular eukaryotic organisms need to have everything they need to survive all contained within that one cell. That means they need to be able to adapt to changes in their environment. They need to be able to have organelles that are going to help them process different kinds of food or handle different kind of stressors in their environment, like say pollution that might be in a pond, or changes in how much sunlight they're exposed to, or how much oxygen they're exposed to. So all of these changes in their environment, they have to be able to adapt to. And so they need those excess organelles because they help with that adaptability. Whereas multicellular organisms are going to be specializing. You're going to have individual cells that perform very specific tasks within the body of that organism. And because they specialize, they can get rid of some of that excess. They don't need all of the tools. They don't need to be adaptable. They just need to be very efficient at their one specific job. Just like how we talked about individual organelles help with specialization within a given cell. Same idea for individual cells within a given body. If you have lots of cells in your body, those cells are going to be different from each other because they're going to specialize for their different jobs. Like here, we've got stem cells and bone cells and blood cells and muscle cells and so on. All of these different kind of cells specialize and adapt to perform their given job within the body. So they can be smaller and they can have fewer organelles within them because they can get rid of the ones they don't need for their job. Then in exercise 5, we look at membranes, and I've already mentioned the makeup of membranes is they're comprised of lipids and proteins. And we said it is semi-permeable, which means it lets some things in and some things out and other things not. And that it's fluid-mosaic, meaning the pieces that make up this membrane can move around. They're not in a rigid set place. And to look at any image of a membrane, you'll want to be able to pick out in that image what structures are the lipids and what structures are the proteins. The lipids are all these line of cells here, or sorry, not cells, all these lines of lipids here that are nice, tightly packed together, that has what looks like a head with two tails. We call the head hydrophilic and the tail's hydrophobic. And that helps it maintain the shape that it needs. The hydrophilic side is attracted to water, and the hydrophobic side doesn't like water. Think of trying to mix oil with water, right? Oils have lipids in there, and lipids are an oily substance, a fatty, oily substance. And so that part that doesn't like to interact with water is this part that's the hydrophobic side. It's phobic, think afraid or don't like or not attracted to it. So it's going to be away from the water. And we call it a bilayer, phospholipid bilayer, because there's two sides. You're going to have some of these lipids on one side and some on the other, with the heads pointing out towards either the inner portion of the cell or the extracellular fluid, and those tails are going to be pointing towards each other. So they're avoiding touching any liquid because they don't like the water. And then interspersed between all of these lipids, you're going to find some proteins. And there's lots of different kinds of proteins within cells. Some of them form channels, like this here kind of shows you a protein that looks like it's shaped like a tube. And so those channels allow things to pass through them. So they'll be selective. They'll have a shape that's specific to certain types of molecules that it wants to let through. And there's going to be proteins like these that have these kind of chains or tails coming off of them. Those are spaces where they have receptors for something. So if something's coming along in this extracellular fluid that will attach to this tail, then that signals a reaction that might open a channel that was closed, or that might close a channel that was open, or other things that might happen based on that reaction when they touch each other. So you have proteins that form channels, and you have proteins that are essentially like little hands waving, waiting for somebody to come along and shake their hand and be like, oh, yes, now we're going to open this door to let you in, or we're going to close this door because we don't want you to come in. So these proteins that are interspersed along the membrane essentially assist with letting things in and out of that membrane. And then in the experiment that you look at in exercise 5, you take a look at these questions about how heat and alcohol affect the integrity of this membrane. And you look at that with some beets. So you should have seen some images of some beet that was cut into little cubes. And it was a nice red color. That red color is there because there's some pigment inside the water that's held in that central vacuole of the beet cells. And if the membrane stays intact and strong and keeps its integrity, then that color, that pigment, stays within the central vacuole and won't escape. And so you have some of that beet put in 3 different test tubes, and those test tubes are put in 3 different conditions. One where it was just cold water, one where it was warm water, and one where it was alcohol instead of water. And you get to see in that experiment what happens to the pigment when those beet cells are exposed to those different conditions. So exercise 6, we start getting into why this size matters. I mentioned it already back in exercise 1. Size is really important to efficiency. And that's because it increases what we call the surface area to volume ratio. That means that the amount of surface area you have compared to your volume is bigger. You have more surface area compared to your volume than you do when you are a bigger cell. So small cells are more efficient at letting things across their surfaces. And this will affect whatever job you have as a cell or as an organelle, because remember, organelles have membranes too and let things pass across those membranes too. So any kind of job that requires things moving across your surface, you're going to be more efficient at with more surface area. So things that might alter your surface area and result in a larger surface area will make you more efficient at that job. But I gave an example here too. There's another job like storage. If your job is storage, you don't want things passing across your surface a lot because you're trying to keep it contained. So things that are storing things are going to want to be as big as possible. So that's why, for example, that large central vacuole in plant cells, that's why it's so large and why it swells up, because it is storing that water. It's trying to be very good at water storage compared to something like the mitochondria that needs to have reactions happen across the surface of its membrane. It's going to want to be smaller and have ways that it increases its surface area to volume ratio to help increase the efficiency of those reactions. So in exercise 6, you do some math where you calculate the surface area and the volume of a cube depending on how big that cube is. And this works for other shapes too, not just cubes. It's just the math is easier with a cube. So they give you that to help make the math nice and easy for you. And of course, when your cube gets bigger, its surface area is going to get bigger and its volume is going to get bigger. But you should see as you do that math that the surface area to volume ratio gets smaller because the surface area gets bigger faster. The volume gets bigger faster than the surface area does. So you end up with more volume compared to your surface area. Then the last exercise of this unit looks at some of these specializations that I mentioned that help increase surface area in organelles that have jobs that require things moving across the surface of their membrane. You look at the mitochondria and chloroplasts and their specializations in this exercise. Both of these are organelles that have two membranes. They have an inner membrane and an outer membrane. And that inner membrane is the one that has these specializations that help it be more efficient at moving things across its surface. And you'll need to be familiar with the terms for those specializations of their inner membranes. In mitochondria, it's called Cristae. And that's those finger-like projections that I described when explaining what the mitochondria do. And in chloroplasts, it's those what looks like a stack of coins, and they call that grana. So in chloroplasts, your specializations are called grana. In mitochondria, it's called cristae. And they look different, but their goal is essentially the same. They're increasing surface area. And that increase of surface area means that reactions can happen faster across those surfaces. Now, this isn't one you necessarily need to know, but it's another example I provide that shows you that this applies to the whole cell too, not just to the organelles. Your different cells that specialize to perform different jobs will sometimes have specializations that also increase the surface area of the whole cell. And that's really very easily seen with neurons, because neurons have what we call dendrites, which, here's another image of a neuron I used here. This is the main cell body of the neuron, and all of these projections coming off of the cell are dendrites. And that's part of the cell. And you can imagine the surface area of these nice long tube-like portions is going to be much greater than if the cell had just been a whole circle. So it's increased its surface area. And that's important for neurons because the process that sends a signal from one neuron to another requires ions passing across the membrane that result in a change in the charge. So it's sending an electrical signal by changing the charge. So it has positively charged ions that pass in and out, resulting in that charge passing along the dendrite. And so this electrical charge then transfers from one neuron to another neuron. And so it's able to send these signals very, very quickly, right? Which is extremely important because your nervous system needs you to be able to react quickly to stimuli that might be dangerous for you, like when something is hot and you don't want to burn your finger. You need to be able to very quickly pull your hand back away from that thing that is hot. So this is really important in neurons and in these organelles that you need to be familiar with. So mitochondria have these cristae because they are making the energy your cell needs. So you can't run out of that energy or your cells are going to die. So that's really critical. And the chloroplasts in plants are also involved in this process because they're taking energy that's in wavelength form from the sun and turning it into the sugars that then get used by the mitochondria to make ATP. So also something that needs to happen nice and quickly so that these plants can have lots and lots of energy to use. So all of this is going to be different pieces that you need to be familiar with in cells. So all of these different organelles, you need to be able to identify the specializations in mitochondria and chloroplasts that alter surface area to volume ratio. Why is that surface area to volume ratio so important? Because that size affects efficiency, and being able to distinguish differences between plant and animal cells, and between prokaryotic and eukaryotic cells, and between single cellular organisms and multicellular organisms. And you can look through the PowerPoint that will be attached that will go through all of the learning objectives for this unit in more detail. But essentially everything I just talked about is going to be part of the learning objectives of this lab and some of the vocab from this lab that you need to be familiar with. And that is Unit 2.